Hello everyone, so happy you could join me for this lecture. It will cover the respiratory system. Let's go ahead and get started. The first thing that we're going to cover is going to be all the primary structures that are going to be used to acquire oxygen and remove carbon dioxide from the blood. There are 7 primary structures: the external nose, the nasal cavity, the pharynx, the larynx, the trachea, bronchi, and then the lungs. Moving into the respiratory system, we're going to start with some broad aspects of respiration, and this is going to be maybe slightly confusing because there's a lot of classifications that we're going to talk about in respect to the various functions of the respiratory system. Starting out, we need to go over these 2 terms, ventilation and respiration. Ventilation is going to be the movement of air into and out of the lungs, while respiration is going to have more to do with the diffusion of gases across the plasma membrane. And there are 2 types of respiration. The first is external respiration, which can also be called pulmonary respiration because this is going to be where you have gas exchange between air in the lungs and blood. So make a little note that external respiration is occurring in the lungs themselves. While the second type of respiration, internal respiration or systemic respiration, is going to be gas exchange between blood and your bodily tissues. So your internal respiration is going to be occurring somewhere in your tissues that are outside of your lungs. So you can think of anywhere where you have capillaries going on in the various tissues of your body. Ventilation and respiration are going to occur in different locations. We are going to classify these locations and sort of functions in 2 different ways. 1, anatomically, and 2, physiologically. So anatomically, we're going to classify these as being the upper and lower respiratory tracts. The upper respiratory tract is going to include the structures from your nose to your larynx, and the lower respiratory tract is going to be the structures from your trachea to the very smallest structure within your lungs, which is the alveoli. The other way that we're classifying, again, is physiologically, and this is going to be the conducting zone and the respiratory zone. The conducting zone is going to be that area that you conduct air into these areas. This is going to include from the nose to these small air tubes in your lungs, strictly for ventilation, and the respiratory zone is going to be from your tubes and alveoli, and this is where gas exchange is occurring. So the conducting zone is pulling air into those areas. The respiratory zone is where gas exchange is actually occurring. Here we have a lovely illustration that is showing the difference between your upper respiratory tract and your lower respiratory tract. We have gone through quite a few of these structures when it comes to lab, so if you need to kind of go through them again, here they are. So moving into the main function of the respiratory system, you can count this as being the numero uno, number 1 function, and that is to accomplish gas exchange. This function is so important that we are going to slap on some processes that occur to accomplish this. So the first is going to be ventilation, which you can just think of as just regular old breathing, air moving in and out of your body. The second is pulmonary respiration, and that is gas diffusion across the alveoli. So oxygen and carbon dioxide primarily are going to be moving into the blood as oxygen, and carbon dioxide is going to be moving out of the blood. The third is going to be gas transport, and this is just how gas is going to be traveling in the blood to and from your cells. And then the fourth is systemic respiration, where we have gas exchange at a tissue level, oxygen moving into the cells and carbon dioxide moving out of the cells. The other functions that we have involving the respiratory system include the regulation of blood pH, so changing your blood carbon dioxide levels is going to change how acidic or basic the blood is going to be. The production of chemical mediators, so this is going to include something called ACE, which is an enzyme involved in blood pressure regulation. Voice production, so the movement of air past vocal folds, making sound and speech. Olfaction, which is going to be your sense of smell. And the last is protection, and this is going to be specifically protection against microorganisms by preventing entry and removing them from the respiratory surfaces, and we'll talk about how that happens in just a bit. Now, we do have quite a few structures and a little bit of the histology to go with them, and we will talk about a lot of structures kind of separately, but these are a few that I do want you to know as far as lecture goes, but primarily you're going to need to know these mostly for lab. So it is talking about your nasal cavity, and anything we've kind of talked about more exclusively with lab, I might just sort of gloss over here and maybe purposefully more talk about the functions rather than the actual anatomy. So when it comes to the external nose, this is everything that's kind of included in it. Again, we have talked about these. The only little difference I want to make is that spelling of conchae. Those are just those little shelves that are in the nasal cavity. So don't let that spelling confuse you. It's the same thing that we talked about in lab, just slightly spelled different. But do know these functions of the nasal cavity and everything kind of are included in the nasal cavity and external nose. And that it is a passageway for air. It's going to clean, humidify, and warm the air that you are breathing in as well as breathing out. It is going to contain olfactory epithelium to help with your sense of smell, and your paranasal sinuses are resonating chambers for speech. All right, so one of the structures that I want to talk about just by itself is going to be the pharynx. This is just a common opening for digestive and respiratory systems. Remember, when we talk about the pharynx, we're talking about the throat, and we have talked about these three regions already. So I am just going to kind of leave this as is. If you want to sit here and read through it, you may, but we have talked about these pretty extensively already. Again, another really lovely picture illustration that is showing all the various structures that you need to know, one for lab, and two, you should in general know what they are already from lab, but also for lecture. Okay. Moving on from the pharynx to the larynx. The larynx is going to be essentially your voice box. It's going to house ligaments used for speech, and it has these various cartilages that you need to know. Again, we've talked about this in lab, so I'm just going to kind of gloss over it. Another lovely illustration showing the various cartilages within the larynx, as well as the various structures you can see in the larynx. The functions of the larynx are going to include maintaining an open passageway for air movement. Remember, with that cartilage, as well as those cartilaginous rings that surround the trachea, that is going to keep the airway open, right? You have the epiglottis and the vestibular folds, which are going to prevent swallowed material from moving into the larynx and thus into the trachea and possibly further down, which wouldn't be so good, into the primary bronchi. So that is something that is helping you not to asphyxiate on whatever it may be, whether it's liquid or food. Your vocal folds are going to be the primary source of sound production. The greater the amplitude of vibration, the louder the sound will be. The frequency of vibration is going to determine the pitch, And then our arytenoid cartilages and skeletal muscles determine the length of vocal folds and also abduct the folds when not speaking to pull them out of the way, which is going to make the glottis larger and allow for more air to pass through. And the fourth function of the larynx is the pseudostratified ciliated columnar epithelium is going to trap debris. And remember, it's got those lovely little goblet cells that are going to produce that nice sticky mucus, which is going to trap that debris. And then those cilia are going to be moving and pushing the mucus and debris out, which we kind of call phlegm, right? And either you're going to cough it up or you're going to swallow it. So here we have another lovely illustration and then a picture of an actual larynx, which is really cool. And you can see how these various movements are going to change how the structure functions. Also, you can see how close your tongue is to, or like the root of your tongue more so, is to the larynx, which is kind of neat and weird to think about. Moving on to the trachea. So the trachea is going to be what you can kind of think of as your windpipe. It is a membranous tube of dense regular connective tissue and smooth muscle. It is supported by 15 to 20 hyaline cartilage C-shaped rings that are open on the posterior side. Remember, we looked at these on the models as well as in that little histology sliced area, right? So it is going to be this C-shaped ring that is open in the back. But again, the purpose of these are going to be to hold open the trachea so that air can always pass through, unlike the esophagus, which can collapse. Our inner lining of our trachea is going to be pseudostratified ciliated columnar epithelium with those, again, I keep calling them lovely because I love goblet cells, and then how they function. And then we do know that the trachea is going to split, and where it bifurcates, so splits, is going to be called the carina. It is going to split into the left and right primary bronchi. Here we have a lovely photo showing our tissue within the trachea, as well as all of the lovely tissues associated with the surrounding of the trachea itself. You can also see the esophagus in that top right histology photo, which is kind of cool to see the trachea compared to the esophagus. That trachea, the lumen of it is going to always stay open, while the esophagus can kind of collapse in on itself. All right, moving on to bronchi. So again, our trachea is going to divide into two main bronchi and separate right at that carina. The carina is very sensitive to mechanical stimulation, so anything that you might inspire, so something that you might inhale, maybe like a piece of popcorn or like a peanut or a candy, or I used to choke on ice all the time for some reason when I was a kid. But some type of material that you kind of asphyxiate on, if it hits that carina, it's going to stimulate a powerful cough reflex. So that's why you kind of cough things up really hard if you swallow it down the wrong pipe. Okay, moving on to the tracheobronchial tree. Remember in lab, I kind of kept comparing the respiratory system to a tree itself, where the trachea is that primary trunk, and then as the structures move out from the trachea, they get smaller and smaller. So here is where we're kind of talking about that. So what they're calling it is the 4 classes of passages. That should say largest to smallest, so we're just going from the larger structures to the smaller structures here. So you have the lobar, or what we call in lab the secondary bronchi. The segmental or tertiary bronchi just continues to get smaller there and kind of splitting off, branching off. Then we have the bronchioles, which are going to be very small, and last but not least, the terminal bronchioles, which will then interact more closely with your alveoli. We do have this lovely illustration and a x-ray of a tracheobronchial tree within one lung. So kind of cool. So again, just keep in mind, just like a tree, moving from the trunk all the way out to the ends where you have the leaves, which in this case I'd consider the alveoli like leaves. You have these bronchi and bronchioles, which are going to be like the limbs and twigs and little sticks that make up that tree. All right, so moving on to changes in those little tree limbs, if you will, which is going to change how much air can pass through. So our bronchi and bronchioles are capable of changing their diameter. If they're going to relax and essentially just increase our airflow, that is going to be called bronchodilation. So those tubes are going to open up and allow more air to pass through. Contrary to that, we have bronchoconstriction, where that smooth muscle will contract and decrease the amount of air able to pass through. What this can also be called in some instances is going to be an asthma attack. And if you have asthma, this is not a fun feeling to have when it occurs, but typically it's going to be an inflammatory reaction that causes severe bronchoconstriction. How this occurs is due to some chemicals called leukotrienes and things that can help are medicines such as albuterol. This is going to cause the smooth muscle to relax in the walls of the terminal bronchioles. Another thing that can cause problems in bronchoconstriction is emphysema. So this is damaged bronchioles collapsing. And what happens is we're not getting enough air moving through. And essentially you're having kind of dead air in the lungs, which is not a good time. But that is something that can be caused by smoking. So moving on to the smallest of our structures when it comes to the respiratory system is our sweet little alveoli. And remember, I kind of compare them to leaves. But this is going to be where we have pulmonary respiration occurring. So that exchange of gases between the structures of the lungs and your blood. And this is going to be occurring strictly in your lungs. So branching from terminal bronchioles to alveoli, we are going to have respiratory bronchioles, and then alveolar ducts, and then our alveolar sacs, which are going to be made up of individual alveoli. In the alveoli, we are not going to have any cilia, but debris is going to be removed by macrophages thanks to our lymphatic system. And macrophages then are going to move into nearby lymphatic vessels or possibly into the terminal bronchioles to be coughed out and up. So here we have that illustration showing our bronchioles moving into an alveolar duct and thus into an alveolar sac. And you can see each of these individual alveoli kind of looking like grapes. So altogether, an alveolar sac ends up looking kind of like a cluster of grapes. And do keep in mind that if we cut that in half, it does have a tendency to just be like an open area. So it's not like each alveoli is completely individualized. They all kind of clump together to make a big sac, if you will, an open area. We do have our lovely little histology slide here showing our alveoli. So this is a good reference picture when it comes to lab. Okay, let's move on to alveolar structure. So in your lungs, you have about 300 million alveoli together between your two lungs, and there are two types of cells from your alveolar wall. And do remember that these walls are going to be very, very thin. And that is going to help with that gas exchange occurring. So type 1 pneumocytes Remember, I'm human and I always mess up words. So type 1 pneumocytes, these are going to be thin squamous epithelial cells that form 90% of the surface alveolus. And do underline this. This is where we're going to have gas exchange happening. So specifically gas exchange is our function here. Type 2 pneumocytes are going to be round or cube-shaped secretory cells, and they are going to produce something called surfactant. So they are secreting surfactant. And we'll talk about surfactant a little bit more later, but just know that with surfactant, it's going to make it easier for our alveoli to expand during inspiration. So continuing on with the alveolar structure, we're going to talk about the alveolar walls and the surrounding pulmonary capillaries, which are just called the respiratory membrane. The respiratory membrane is going to be the location of pulmonary respiration. It is where oxygen enters the blood and carbon dioxide exits the blood and into the lungs to be expired. The membrane is going to be very, very thin and composed of alveolar cell layer, capillary endothelial layer, and interstitial space. Do keep in mind, I know I said that it's thin, but just to give you an idea, it's thinner than a single piece of tissue paper, so super duper thin, and it is composed of 6 layers, and you can read through those if you'd like, but those are the 6 layers. Going from superficial to deep. Here we have a nice illustration, and I will talk about both of these here. So for the top illustration, where you see that little A, that is a section of an alveolus showing the air-filled interior and thin walls composed of simple squamous epithelium. The alveolus is surrounded by elastic connective tissue and pulmonary capillaries, so it's just showing you those layers there. That second illustration that is kind of showing you more of a close-up going on, it is labeled B, is showing you diffusion of oxygen and carbon dioxide across the 6 thin layers of the respiratory membrane. And so those 6 layers are what we just looked at on that last slide. So moving on from the smaller structures, we're going to look at some bigger structures now in the form of muscles. So these are going to be what make up the thoracic wall, as well as the muscles of respiration. The thoracic wall is composed of 5 layers. You have the thoracic vertebrae, the ribs, costal cartilage, the sternum, and muscles. And I said layers, but they're not really layers, they're structures. It's made up of 5 structures. I should have said structures. But the thoracic cavity is the space enclosed by the thoracic wall and the diaphragm. Remember, the diaphragm is that big muscle that spans right underneath the thoracic cavity, and it's going to separate the thoracic cavity from the abdominal pelvic cavity. Your muscles are going to change your cavity size within the thoracic cavity during ventilation. So that is going to help with changing pressure so that you breathe in or breathe out. Moving on to another very important structure is going to be the lungs. Your lungs are going to be your primary organs of respiration. We have talked about all of this already in lab, so again, glossing over it, but just know the base, the apex, the hilum, and the various lobes. And I told you in lab that they do have various fissures, so little separations between the lobes, but I didn't ask you to know the fissure names, but I do think in this lecture it does kind of go over it on the next slide. Right, so this does show you the lobes and the bronchi-pulmonary segments of the lungs. You don't need to memorize all of these. Just know that they exist and that this graphic at the bottom that's very colorful is just showing the brachiopulmonary segments, and why they're segmented like this is because they are supplied by various segmental bronchi, and so it's separated out by those bronchi, what supplies them, if you will. Okay, so moving away from structures and into blood and lymphatic supply, because obviously the lungs are going to need a lot of both, right? So there are going to be two sources of blood to the lungs, which we have already discussed before in the cardiovascular system. So the pulmonary artery is going to bring deoxygenated blood to the lungs from the right side of the heart to be oxygenated in the capillary beds that surround the alveoli, and then oxygenated blood is then going to travel to the tissue of the bronchi, bronchial arteries, and to the capillaries. And part of this is now deoxygenated blood and exits through the bronchial veins to the azygos, which is a part that merges with blood of alveolar capillaries and returns to the left side of the heart. So again, we have talked about this extensively when it came to the cardiovascular system, so hopefully that's just a little bit of a recap. What should be new is the two lymphatic supplies. So we have the superficial and deep lymphatic vessels, which are going to exit the lungs through the hilus. And remember, the hilus or hilum is going to be that area where you have blood coming in, blood coming out, and then your lymphatic vessels. The superficial lymphatic vessel is going to drain the superficial lung tissue and visceral pleura, and the deep lymphatic vessel is going to drain bronchi and associated connective tissue. There are no lymphatic drains in the alveoli themselves. So a few more things kind of about the lymphatic system being associated with your lungs. One, you have a lot of gas exchange and blood going through there, so you're going to need a drain, because remember, when it comes to the lymphatic system, that's its really big purpose, aside from containing white blood cells and being part of your immune system, right? You need to drain that fluid that is coming out of the blood during gas exchange so that you don't have those fluids clogging up those tissues. So one of the other things with that is you have phagocytic cells within that lymphatic fluid that's going to be there in the lungs, and also associated with those vessels that are going to take away carbon particles and debris. We talked about those before, those macrophages, and move them to lymphatic vessels to be taken out or possibly be coughed out, right? In smokers, the debris is going to be too great for these macrophages to keep up, and will essentially, that debris will stain the lungs. That's why smoker lungs have a tendency to be more on a darker gray color than a nice healthy pink. That debris ends up coating the alveolar surfaces, and because of what's in smoke from cigarettes and things of that nature, they have carcinogens, which are cancer-causing agents, and cancer cells can form in the lungs due to the increased amount of carcinogens coating the inside of the lungs, which, due to the lymphatic system's association with the lungs, the lymphatic system can inadvertently take the cancer from the lungs and transport it throughout the entire body. So that is why lung cancer is so bad, and that's why you shouldn't smoke. Okay, I'll get off my soapbox there and we'll talk about pleura. When it comes to this term pleura, what we're talking about serous membranes. Also, pleura is going to be in reference to your lungs. So the pleural cavities are going to be the surrounding of each lung and is formed by pleural membranes. You have the mediastinum, which is going to be that central cavity that contains contents of the thoracic cavity except for the lungs. It's mainly going to hold your heart. You're going to have a visceral pleura, which is going to be a membrane that is adherent to the lung, so it sticks to the lung. It is a serous membrane. And then you have the parietal pleura, which is going to be adherent to the internal thoracic wall. It's still going to surround the lung, but it's not attached to the lung. The visceral pleura is attached to the lung. The parietal pleura is going to be attached to the thoracic wall. So really the only difference between the visceral and parietal pleura is what it is clinging to. This illustration is showing you a transverse section of the the thorax, and shows the relationship of the pleural cavities to the thoracic organs. So of course you have your lungs, your heart, and those various structures surrounding them. But that's all it's really showing you is those cavities and the pleural membranes. Okay, so we've talked a lot about structures. We're going to start talking about gases because that's one of the big important things. Remember, the main function of the lungs and the respiratory system is going to be for gas exchange. So we are going to first talk about the muscles of respiration that are going to change the volume of thoracic cavity, which is going to allow air to move into and out of the lungs. Various muscles that are going to help in this process are going to include the diaphragm, external intercostals, pectoralis minor, and scalene muscles. Your ribs are going to elevate, so the ribs themselves, the bone, which are going to have muscles attached to them, those intercostals. The ribs are going to elevate like the handle on a bucket, which is going to kind of pull up and out. And there's a illustration on the next slide that's going to show that a little bit easier. So just kind of think of it like that. Your diaphragm itself is going to be a dome-shaped muscle, and we've seen it before. You've seen it in the cats, and it's going to have a central tendon attached to it. And those are the muscles of inspiration. The muscles of expiration are going to decrease the volume of the cavity. So that's the biggest important thing. Inspiration is going to increase the volume of the cavity. Expiration is going to decrease the volume of the cavity. If you need to remember it this way, I inspiration, I increase, and I don't know how you remember expiration other than it's the opposite of inspiration. But the muscles associated with expiration are going to be the internal intercostals, transverse thoracic, and assistance from your abdominal muscles. So here we are looking at, on the left side, the elevation of the ribs in the bucket handle movement. It's going to increase your thoracic volume laterally. On the other side, we have, as the rib is elevated, rotation of the rib in the pump handle movement, different than the bucket handle, is going to increase thoracic volume anteriorly, so as opposed to laterally in the other way. Okay, now you guys' favorite part. I know it's going to be kind of annoying to kind of think about and look at, but I'm not going to ask you to memorize all of it, and I'm not going to ask you to give me an example of any of these. I just want you to know in general what these equations are going to represent and the relationships that they look at. So the first thing we're going to do is look at the relationship between pressure gradients and ventilation. And do keep in mind that this is going to be an inverse relationship. So as pressure increases, ventilation is going to, volume. Too many words that start with V. So let me start that over. Pressure and volume. This is going to have an inverse relationship. Pressure is going to increase while volume is going to decrease. So that is an inverse relationship. What we are looking at first is going to be something called Boyle's law. Boyle's law is that states that the pressure of a gas in a container at a constant temperature. So this constant temperature that we're going to be talking about, which is represented by the letter K, in this case is going to be body temperature. It can be considered a constant. So as the volume of a container increases, for example, your thoracic cavity during inspiration, the pressure inside will decrease, and then vice versa. Moving on to pressure gradients and airflows, the airflow is going to go down its pressure gradient from higher pressure to lower pressure. Underline that, highlight it, tattoo on your arm, whatever you need to. When it comes to everything that we're talking about through gas exchange, when it comes to the respiratory system, as well as when we were talking about the cardiovascular system, we are going to be moving down the pressure gradient. We're not going to be moving against it. We want to go with the flow, if you will. That means that we are going to be moving from high to low, in this case, pressure. So we are just moving down that gradient. And the greater the difference in pressure, so if it's a really high pressure going to a really low pressure, it is going to be a faster flow rate. And so that is what we're kind of looking at, is airflow or flow rate with pressure gradients. And I do have a little fun fact. A decrease in pressure gradient between atmospheric air and your alveoli at higher altitudes is why it makes it feel like it's harder to breathe. And that is just due to that really large difference in pressure gradient. So kind of cool, fun fact. Okay, so there are several types of tests that can be performed if your respiratory system is malfunctioning, and this is one of the ways that we can measure that. So you would use something called a spirometer. Spirometry itself is going to measure volumes of air that move into and out of the respiratory system. We are going to use these 4 ways or 4 different pulmonary volumes in order to kind of quantify our pulmonary volumes. So the first is going to be tidal volume. Your tidal volume is going to be the amount of air inspired or expired with each breath. At rest, it's about 500 milliliters. And this will increase with activity. So this, again, that is at rest, but it will increase quite a bit if you are being physically active. You then have your inspiratory reserve volume. This is the amount that can be inspired forcefully after inspiration of the tidal volume. It's about 3,000 milliliters at rest. You have the expiratory reserve volume, which is the amount that can be forcefully expired after expiration of the tidal volume. That's about 1,100 milliliters at rest. And then you have residual volume, which is the volume still remaining in the respiratory passages and lungs after the most forceful expiration you can muster at 1,200 milliliters. Continuing on with ways that we measure lung function, we have something called pulmonary capacity. Pulmonary capacity is going to be the sum of 2 or more pulmonary volumes. And those pulmonary volumes, some examples include inspiratory capacity, which is going to be your tidal volume plus the inspiratory reserve volume. So we're just combining those two. Again, the pulmonary capacities are going to be the sum of 2 or more pulmonary volumes. So all of these are just going to be something plus another something, right? The next is going to be the functional residual capacity, which is going to be your expiratory reserve volume plus your residual volume. Vital capacity, which is the sum of your inspiratory reserve volume, tidal volume, and expiratory reserve volume. So that includes 3, the sum of 3 different measurements. And then you have your total lung capacity, which is going to be the sum of inspiratory and expiratory reserve volumes plus your tidal volume and residual volume. Okay, so this little graph is going to show your lung volumes and lung capacities, and it is in general just showing you the tidal volume during resting conditions. I'm not going to ask anything from this, but it is kind of nice to see that visual there if you needed a visual to go with the various measurements of lung function. This is the last slide talking about measurement of lung function. And this has a very important vital sign that healthcare workers will look at. Probably one of the first things they look at, aside from like blood pressure, is going to be respiratory rate. And that is the number of breaths taken per minute. We are going to talk about minute volume, which is going to be the total air moved into and out of the respiratory system each minute. And how we get that is like tidal volume times respiratory rate. And I am going to give you a little example here just so you can kind of look at that and see how it would go. So let me get this little guy here. Okay, so my example of minute volume, and pardon the bad handwriting, I can't really control this tablet as well as I'd like. So our tidal volume we're going to say is 500 milliliters, and we're going to multiply that by our respiratory rate, which we're going to call it 12 breaths a minute. So if we do the math there, that's going to give us 6,000 milliliters a minute, or you can kind of just convert it into 6 liters a minute. And that is how we would get our minute volume there. One thing that minute volume does not measure is the amount of air available for gas exchange. Minute volume is just an indication of your carbon dioxide levels. What does measure the volume of air available for gas exchange per minute is going to be alveolar ventilation. And then we have only a portion of each breath reaching the alveoli. The remaining areas where no exchange occurs is going to be called dead space. So that is where we are going to start talking about anatomical dead space, which is going to be formed by the nasal cavity, pharynx, larynx, trachea, bronchi, bronchioles, and terminal bronchioles. You have the physiological dead space, which is the anatomic dead space plus the volume of any alveoli in which gas exchange is less than normal. And then you have something called alveolar ventilation, represented by a V and a subscript A, where the volume of air, it is the volume of air available for gas exchange per minute. So some factors that can affect ventilation include increased vital capacities and correlate with male gender, height, being active, being slender, and being healthy, and that is not in a disease state. So disease states will increase that more than just these in general statements. So an example would be your vital capacity for adult females is actually 20 to 25% less than adult males. Another example is that in tall people, you have a greater vital capacity than in someone who is height challenged, if you will. Do you know that the vital capacity reaches the max amount in young adults, and it will immediately decrease as you get older? So we've talked about ventilation supplying atmospheric air to the alveoli. Next up is going to be pulmonary respiration, which is the diffusion of gases between alveoli and your blood. So we are still moving down that pressure gradient when we're talking about these various things. So do keep that in mind as we go through. So one of the big things that we're going to be looking at are a few concepts to understand the movement of gases. So the first thing that we need to discuss is going to be partial pressure. So this I want you to think of as the individual pressure of each gas that we're going to be talking about. In this case, we're going to be looking at nitrogen, oxygen, carbon dioxide, and water vapor. And I'll tell you what those numbers are here in just a second. But we do need to look at Dalton's law real quick. Dalton's law is going to be the total pressure being the sum of the individual pressures of each gas. Okay, so from there, and if you want to write these down, you can, you do not have to, but just to give you an idea of these partial pressures of these various gases that I mentioned before. Nitrogen is 600 millimeters of mercury, while oxygen is 160 millimeters of mercury. Carbon dioxide is 0.3 millimeters of mercury, and water vapor is 0. So, kind of interesting to see all of those in comparison. And remember, that is just a partial pressure. It is the amount of pressure exerted by each type of gas in a mixture. Specifically, what we're looking at in this case is going to be a liquid. So, diffusion of gases through liquids, that is going to be called sorry guys. So the diffusion of gases through liquids. This is going to be how gases move from partial pressure to lower partial pressure. And when the partial pressure of gases are equal between air and liquid, this is going to be equilibrium, which is what we call Henry's law. So again, keep in mind we were talking about the diffusion of gases through liquids. And then the last thing on here is the diffusion coefficient. This is the rate at which gas diffuses into and out of a liquid or a tissue. And factors involved in determining this rate are going to be your solubility coefficient and molecular weight of the gas that you're looking at specifically. Next up, we have the step-by-step mechanism of alveolar respiration. So you do have these various mechanisms of alveolar ventilation, which is going to be the barometric air pressure, which is the air pressure outside of your body, and then you have intraalveolar pressure, which obviously is air pressure within your alveoli. So moving into those steps, we have the alveolar ventilation during quiet resting and kind of that whole process there. Now, I am not going to go over this completely, but do make it a little note that we are talking about it where it is alveolar ventilation when you are in a resting state. On the next slide, it does have these 4 steps illustrated, so we're going to move on to that instead of going through it on this slide. Okay, so the combined space, something that you should know about the slide, excuse me, the combined space of all the alveoli are represented by a large bubble represented in blue. And you can see it getting bigger between stages 1, 2, and 3. And then it kind of shrinks down a little bit on step number 4. But the alveoli are actually microscopic, and you wouldn't be able to view it. This is just a nice little exaggerated illustration so that you can see it kind of on this bigger scale, even though technically you wouldn't. So yeah, those are the 4 steps of alveolar respiration. So some factors affecting alveolar respiration, the first being lung recoil. This is going to be the tendency for lungs to decrease in size after being stretched due to elastic recoil and surface tension. So what those terms mean, elastic recoil, is the elastic fibers in the alveolar walls return to the original shape after being stretched, and surface tension is the film of fluid that lines the alveoli. Where water interfaces with air, polar water molecules have great attraction to each other with a net pull in toward other water molecules. This is going to tend to make alveoli collapse. And as I promised before, we were going to talk about surfactant again. Remember that surfactant is going to be a secretion. What it does is it prevents the tendency of lungs to collapse by reducing surface tension, and it is produced by type 2 pneumocytes, which we talked about before. The term surfactant is actually just a combination of 3 words, which is, once you hear it, it sounds really silly. Surface acting agent. They just squished all those words together, took out a few letters, and said, we'll call it surfactant. I love it. So it is 1 molecule thick as a layer. So it's very, very thin over the alveolar fluid, which is going to reduce that surface tension. With surfactant, your surface tension is 3 millimeters of mercury. And without it, it's 30 millimeters of mercury. So it's very important. And what it is going to help prevent is your lungs collapsing. So you need that surfactant to help make sure that your lungs do not collapse. You have less of a tendency to have your lungs collapse. Continuing on, we'll talk about pleural pressure, and that is one of the factors affecting alveolar ventilation. Pleural pressure is a pressure within the pleural cavity between the parietal and visceral pleura. Remember those two membranes that are going to be either surrounding and attached to the lungs, or it's going to be attached to the thoracic wall. Yeah. So negative pressure can cause lungs to expand during inspiration, and the alveoli expand when pleural pressure is low enough to overcome lung recoil. Another factor that can affect alveolar ventilation is going to be a pneumothorax, which is an opening between the pleural cavity and air that is going to cause a loss of pleural pressure. If the visceral and parietal pleurae become separated, if the thoracic cavity is pierced, essentially, then the lung will collapse. But because there are two separate pleural cavities, there's a good chance only one lung collapses, and you'll be kind of not okay, but more okay than if they both collapse, right? So this is a really common thing to see in a lot of medical shows, at least from the ones I've seen, Grey's Anatomy, The Good Doctor, ER, oh my gosh, so many in ER. But it's where, you know, someone sticks like a pin cap into someone's side between two ribs, and then suddenly the person can like breathe so easily because they basically overcame that pneumothorax, you know. But that's at least what I always think of when I think of that. Okay, so we have the dynamics of normal breathing cycle as a little graphic here. So during inspiration, air flows into the lungs because the pressure in the thoracic cavity decreases. During expiration, air flows out of the lungs because the pressure in the thoracic cavity increases, and that is what this little graphic is showing us. Okay, so some factors affecting diffusion through the respiratory membrane. Diffusion of gases through the respiratory membrane depends on three major factors. The first is partial pressure gradients, and we've talked about what partial pressure is. The second is membrane thickness. The thicker the respiratory membrane, the lower the diffusion rate will be, which makes sense because it's just going to be harder for those gases to be able to go through. And then the surface area, and decreased surface area is going to decrease the diffusion rate. So typically whenever you increase surface area, you're going to be increasing whatever mechanism it is that you are trying to accomplish, which makes sense. Okay, so this slide is a nice little illustration going along with some steps that is showing you how gas exchange occurs. Partial pressure gradients of oxygen and carbon dioxide between the alveoli and the pulmonary capillaries, and between the tissues and... I'll be honest, my cat just jumped onto my table and I thought I'd leave it in there because there's two slides I can kind of go through this information. Oh my goodness. Get down. I'm so sorry, he's being such a brat. Okay. So we are looking at gas exchange when it comes to this graphic, and you can go and read through each of the little steps where it has the PO2 and PCO2. That is just talking about partial pressure. Okay, so what this illustration is showing us is going to be the partial pressure gradients of oxygen and carbon dioxide between the alveoli and the pulmonary capillaries, and between the tissues and the tissue capillaries are responsible for gas exchange, and all partial pressures shown are expressed in millimeters of mercury. So this is just going through giving you some numbers that you can kind of look at. You don't need to memorize them, please don't. But if you can just go through, look at it, and kind of understand what it's kind of going through and telling you when it's showing you those numbers, that's all I really want you to get. Just understand the basic concept would be fine. Okay, so now that we've kind of looked at, in general, gas exchange, let's start talking about transportation of these gases, specifically oxygen and carbon dioxide. So how this is going to occur is one of the things is going to be hemoglobin. Hemoglobin, we talked about that before. Remember, it's the 4 chains of heme with a other way around. 4 chains of globin with a heme group in the middle. And that heme group is going to have an iron molecule, which that iron molecule can then attach to 2 oxygen molecules, right? So for each hemoglobin, you can hold about 8 oxygen molecules at capacity. And hemoglobin is going to be that protein that makes up red blood cells. So there are 4 types of hemoglobin, embryonic and fetal, which are going to be exclusively the type of hemoglobin that is going to be at those stages of development. More commonly is adult hemoglobin, and this is essentially the type of hemoglobin that we were discussing in chapter 19 when we were talking about blood. And then the last is going to be hemoglobin S, which is going to be a type of hemoglobin that is found in individuals suffering from sickle cell disease. So, transporting oxygen, hemoglobin is going to be transporting, oxygen is transported by hemoglobin. About 98.5% of the oxygen is going to be transported by hemoglobin, and then only about 1.5% is going to be oxygen that is dissolved in your plasma. So, starting with the transport of carbon dioxide, this is specifically a byproduct of the breakdown of glucose when cells use oxygen to produce ATP. That is going to create carbon dioxide. So that is a waste product. Carbon dioxide can be transported in 3 ways. Dissolved in plasma, that's about 7%. Carbon dioxide is going to be doing that. And the second is about 23% is going to be bound to hemoglobin and transported. And the third is about 70%, so what's left is converted to bicarbonate ions. And the reason why we are transporting carbon dioxide, it's very important, is that too much carbon dioxide is going to cause the pH in our blood to decrease and thus become more acidic. So if you were watching the title slide, and you could go back and come back to this one, it's kind of funny how the manufacturer decided to do the titles of these slides. So the first one was transport of carbon dioxide, and then this one is just carbon dioxide transport. Like, okie doke, I'm loving it. Anyway, at a tissue level, as carbon dioxide enters red blood cells, it will react with water to form bicarbonate and hydrogen ions. The chloride ions are going to enter the red blood cells, and bicarbonate ions are going to leave, which is called a chloride shift. Hydrogen ions are going to combine with hemoglobin, lowering the concentration of bicarbonate and hydrogen ions inside the red blood cells, which is then going to promote the conversion of carbon dioxide to bicarbonate ion. And I do believe that in the next few slides, we are going to see this process essentially illustrated out. At pulmonary capillaries, carbon dioxide is going to leave red blood cells, resulting in the formation of additional carbon dioxide from carbonic acid. The bicarbonate ions are going to be exchanged for chloride ions, and the hydrogen ions are going to be released from hemoglobin. So in this illustration, it's just showing you from the start of cellular respiration, producing carbon dioxide as a byproduct, kind of the process of how transportation of carbon dioxide can go. So that first little bit you see is 7% dissolved in plasma, so that's just out in the plasma. But if 93% go into our red blood cells, it's either going to be split up between 23% are going to be bound to hemoglobin, and the other 70% are going to be converted into bicarbonate ions. So this is just showing those 3 different ways that we transport carbon dioxide. This slide is illustrating and kind of writing out how gas exchange is going to occur in your tissues. So in the tissues, the carbon dioxide is going to diffuse into red blood cells where the enzyme carbonic anhydrase is located. Carbonic anhydrase is going to catalyze the reaction of carbon dioxide with water, or H2O, to form carbonic acid, which then is going to dissociate to form bicarbonate ions and hydrogen ions. From there, oxygen will be released from hemoglobin and diffuse into tissue cells. And that is what we are seeing there, starting with step 1 down through step 5. Now, this is going to be essentially just that process that we were looking at before, and it just sort of looks like it's in reverse, really, because instead of oxygen going out, oxygen is coming in. So this is still gas exchange, but this is when it is occurring in the lungs. So in the lungs, carbon dioxide diffuses from red blood cells into the alveoli. Carbonic anhydrase is going to catalyze the formation of carbon dioxide and water, or H2O, from carbonic acid, sorry, end of a sentence. Bicarbonate and hydrogen ions will then combine to replace the carbonic acid, and oxygen diffuses into the red blood cells and binds to hemoglobin. So this is just a similar process, just kind of backwards. Okay, so if you recall, the functions of the respiratory system include maintaining blood oxygen and carbon dioxide concentrations and blood pH. So what we're talking about here is the specialized cells that are going to detect changes in levels out of normal ranges of our pH and oxygen and carbon dioxide levels. So what these are called are chemoreceptors. And these chemoreceptors are very, very important, especially in their locations. So you do have central chemoreceptors that are located in chemosensitive areas of the medulla oblongata. Underline that, highlight that. The medulla oblongata is one of the most important structures when it comes to helping to fix oxygen and carbon dioxide related problems in the blood. And then we have the peripheral chemoreceptors, which are going to be found near the carotid and aortic bodies, so pretty close to those primary arteries, right? Okay, so we are going to look at the effect of partial pressure of oxygen on oxygen transport. Specifically, we're going to look at something called the oxygen hemoglobin dissociation curve. This is going to be essentially a graph that describes the percent saturation of hemoglobin at different partial pressure oxygen values. So what this is going to be doing is if we have a partial pressure of oxygen of 104 millimeters of mercury, then your hemoglobin saturation is going to be at 98%. Now, if we drop the partial pressure of oxygen to 60 millimeters of mercury, your hemoglobin, the saturation level, is only going to go down by 8%. It's still at 90. So due to an affinity, or if instead of affinity you want to think attraction of hemoglobin for oxygen, because hemoglobin has a big crush on oxygen, if you want to think of it that way. It will take as much oxygen as it can, no matter what, essentially. So due to that attraction of hemoglobin for oxygen over a wide range of values, that saturation level is not going to change very much. And do make a little note that that first little bit, so at a partial pressure of 104 millimeters of mercury, that is the normal partial pressure of oxygen in blood leaving the lungs. So that is a very normal area to be in. So when there is an average of 2 oxygen molecules bound to each hemoglobin molecule. Hemoglobin is going to be about 50% saturated. And if you think about it this way, hemoglobin can hold 8 oxygen molecules, right? So if it's only holding 2 at a time and your saturation level is still 50%, that's still pretty good, right? Okay, so we do have this illustration and graph going hand in hand, showing that oxygen hemoglobin dissociation curve. So it is showing the percent saturation of hemoglobin as a function of partial pressure oxygen, the ability of hemoglobin to pick up oxygen in the lungs and release it in the tissues is not dissimilar to a glass filling and emptying. So that's what it's showing you with each percent of oxygen saturation over on the left, and in comparison to where it is with the partial pressure of oxygen on the bottom. So things that can change that curve for the oxygen hemoglobin dissociation curve. The first one we're going to look at is going to be pH, which the effect of pH on oxygen hemoglobin dissociation curve. As pH of blood declines, the amount of oxygen bound to hemoglobin at any given partial pressure oxygen will also decline. What we call this is the Bohr effect. It is going to occur because decreased pH is going to yield increase in hydrogen ions that combines with hemoglobin, changing its shape and oxygen to where it cannot combine. It cannot bind to hemoglobin. Next up is the effects of carbon dioxide and temperature on that curve. An increase in partial pressure carbon dioxide is going to cause a decrease in pH as well, so essentially that Bohr effect. And carbonic anhydrase is going to cause carbon dioxide and water to combine reversibly and form bicarbonate ions, which ionizes to hydrogen ions and carbonic acid. The increase in temperature is going to decrease the tendency for oxygen to remain bound to hemoglobin. So as temperatures increase in the body, hemoglobin doesn't want to stay stuck to oxygen as much. It doesn't bind as well. So as your metabolism goes up, more oxygen is released into your tissues, which makes sense because chances are your tissues are going to need that oxygen if your body temperature is in fact increasing. So it's just going to start releasing, hemoglobin will start releasing oxygen to fulfill your metabolic needs, if you will. And then the last effect that we're going to look at as far as on that curve is going to be the effect of BPG, which is 2,3-bisofunglycerate. However you want to say it, we'll just say BPG because that's easier, right? This is going to be an effect on oxygen transport that is essentially a byproduct that gets released as from red blood cells as they break down glucose for energy. So not dissimilar to carbon dioxide, but this is something slightly different. So BPG is going to bind to hemoglobin and increase the release of oxygen. Typically, this is going to be in response to low blood oxygen levels. That is the trigger for increasing BPG. So for this first graph, it is showing you the normal curve of the oxygen hemoglobin dissociation curve in red. So that's the curve before the shift. But then we're going to have pH decreasing, so we're getting more acidic. The partial pressure carbon dioxide is going to increase, or temperature increases, whatever is causing that pH to decrease and get more acidic. The curve which again, we're showing you the initial curve in red, is going to shift to the right, which is what is being represented as the curve in blue. This is going to result in an increased release of oxygen. Now, I don't really like this because it gets a little confusing, but what this is showing you is the pH increasing. That last slide was showing us the pH decreasing. Now, what I don't like about this is instead of sticking with the colors that they had on the last slide, they changed it on us. So try to erase what you know from the last slide and look at this with fresh eyes. So the curve before the shift is being represented in blue now. After the shift, where we have pH increasing, our partial pressure of carbon dioxide decreases, our temperature is decreasing, our curve, which is initially in blue, is going to shift to the left, and that is our curve in red. This is going to result in an increased ability of hemoglobin to be able to pick up oxygen. So before we're dropping oxygen off, we have a shift to the right, and if we have a shift to the left, we're going to be able to have hemoglobin pick up oxygen instead of dropping it off. So many factors are going to regulate our respiratory rate, particularly with changes to carbon dioxide levels and pH. And we can kind of classify this first regulating mechanism as being local control. So we have pulmonary capillary perfusion, which is going to be the flow of blood to alveoli through pulmonary capillaries, and ventilation perfusion coupling, which is the relationship between ventilation and alveoli blood flow. Excuse me. And those two go hand in hand. One is describing the relationship. So not 100% of cardiac output is fully saturated with oxygen due to something called a physiological shunt. So not all the blood that goes to your lungs is going to be able to be saturated with a bunch of oxygen, which is okay. You never really expect that anyway. But ventilation perfusion coupling can be disrupted by insufficient blood flow to alveoli or insufficient airflow to alveoli. And regional distribution of blood flow determined primarily by gravity, but can also be determined by alveolar partial pressure oxygen. I'm starting to lose my voice, guys. Getting to the end of this lecture, I'm starting to lose my voice. We're almost done, though. Okay, so that was local control. Moving on to neural control. I said to underline this before, it's very important. I'm going to say it again, but our neural control is going to start in our medulla oblongata. So that is the key player here. It means that the neurons in the medulla oblongata will control the basic rhythm of ventilation through stimulation of muscles of respiration. Whew, gotta get through these last few slides. We're so close. Okay, so you have two groups or centers of neurons. So these are just a collection of neurons associated with the medulla oblongata that are going to help in various ways to have that neural control over your respiration. So the medullary respiratory center is going to be a dorsal group that stimulates the diaphragm, and then the ventral group of this center is going to stimulate the intercostal and abdominal muscles. The pontine respiratory group is going to be involved with switching between inspiration and expiration. Okay, so this slide is still talking about your neural control of respiration. I don't know why there's a number 1 there. Maybe the people who made this were wanting to do number 1, starting inspiration, number 2. We'll just put a number 2 there because we can. Okay, guys. So number 2 and then number 3, I guess. But really, it doesn't really matter if you put numbers, but sure, we're going to put those numbers there. So this is going through the process of generating that rhythmic ventilation using that neural control. So the first is starting inspiration, where your medullary respiratory center neurons are continuously active. The center is going to receive stimulation from receptors, and stimulation, that should be stimulation, from parts of your brain concerned with voluntary respiratory movements and emotion. And then the last thing that's going to happen when you're starting that inspiration, combined input from all sources causes action potentials to stimulate respiratory muscles. The second thing is going to be increasing your inspiration. So more and more neurons are going to be activated, and then the last is going to be stopping inspiration. And so your neurons are going to be stimulating. Neurons are stimulating, also responsible for stopping inspiration, and receive input from pontine group and stretch receptors in your lungs. Inhibitory neurons will be activated, and relaxation of respiratory muscles will result in expiration. Okay, so now we are going to move on to the effect of partial pressure oxygen on your respiratory rate. So specifically, we're going to talk about this term, hypoxia. So hypoxia is going to be a decrease in oxygen levels below their normal values. So, in response to hypoxia, your carotid and aortic body chemoreceptors that we were talking about before are going to respond to, excuse me, that decrease in partial pressure oxygen by increased stimulation of the respiratory center to keep it active despite decreasing oxygen levels. Contrary to that, the effect of partial pressure carbon dioxide on the respiratory rate. So your major regulator of respiration, that's so phrased weird. The major regulator of respiration during rest or exercise. So weird phrasing. So even a small increase in carbon dioxide in blood is going to trigger a large increase in rate and depth of respiration. So there's two definitions that I want you to know associated with this, and that is hypercapnia and hypocapnia. Make sure that you know the difference between them. Hypercapnia is going to be greater than normal amount of carbon dioxide. Hypocapnia is going to be a lower than normal amount of carbon dioxide. Typically, when you see something say hyper, it just means more. Hypo means less. So the chemosensitive area in the medulla oblongata is going to be more important for the regulation of partial pressure carbon dioxide and pH. And your carotid bodies will respond rapidly to changes in blood pH, especially in response to exercise. Moving on to the effect of pH on respiratory rate, chemosensitive areas of the medulla oblongata and carotid and aortic bodies are going to respond to your blood pH changes. Chemosensitive areas are going to respond indirectly through changes in carbon dioxide. Your carotid and aortic bodies will respond directly to pH changes, and a lower pH, so more acidic, is going to trigger an increase in rate and depth of breathing. This is going to reduce carbon dioxide and raise your pH back up to normal. So getting rid of that carbon dioxide is going to change your pH, and also it's going to increase how much oxygen you have. So the cerebral and limbic system control of respiratory rate is the rate and depth of breathing controlled both voluntarily and involuntarily by your cerebral cortex. The rate is going to change due to exercise, and it is controlled by inputs to your respiratory center. The highest level of exercise that can be performed without significant change in blood pH is going to be called the anaerobic threshold. Beyond this level, your pH will drop and will stimulate an increased ventilation. This term apnea is going to be the absence of breathing. This can be voluntary or involuntary. And if you'd like to do a little experiment, I'll start a timer now and try to hold your breath. So, as a period of voluntary apnea increases, a greater and greater urge to breathe will develop. You're probably already feeling that if you took my challenge and started holding your breath. It's been about 20 seconds thereabouts. So typically, People can hold their breath about 30 seconds, possibly a few minutes, depending on if you kind of train for it. You can definitely breathe now if you have been holding your breath, please do. But what happens is, due to a higher partial pressure of carbon dioxide levels in your arterial blood, it is begging you, like those chemoreceptors are going to start begging you to breathe if you're holding your breath. So kind of a little fun fact, a little interesting thing, is that the Guinness World Record holder, Alex Sergey Vendrell, held his breath for 24 minutes and 3 seconds in a pool. So you can train yourself to hold your breath, but, you know, I'm not sure, other than trying to beat Alex Vendrell, what the purpose of that would be, but it's kind of fun. Another thing that can affect your respiratory center is going to be your emotions. For example, hyperventilating, possibly during a panic attack, or gasps of crying can also change your respiratory rate. Okay, so this is going to be a graphic showing the regulation of blood pH. And what this is showing us, starting from step 1, is the blood pH is in a normal range. Step 2 is going to be your blood pH increasing outside its normal range, which is going to disturb homeostasis, so that optimal state of being. The third is the control center for blood pH. The medullary chemoreceptors detect an increase in blood pH. Your blood becomes more basic and responds to the increased pH by signaling a decreased breathing rate. The fourth step is going to be the effectors. The diaphragm and the other respiratory muscles respond by slowing their concentration rate, which lowers the rate of breathing. That fifth step, as a result, more carbon dioxide is retained, which causes pH to drop. Your blood becomes more acidic. The sixth step is blood pH returns to its normal range and homeostasis is maintained. What I want you to do is observe the responses to a decrease in blood pH by following those red arrows. So it's kind of a cool little diagram showing you that regulation of blood pH. So as it increases or decreases and how the body will respond. This illustration is showing a major regulatory mechanism of ventilation. When it comes to those little arrows, the green or the red, it is representing a plus or a minus, which is either the plus being an increase in ventilation or the minus being a decrease in ventilation. So during exercise, respiratory rate changes are controlled through various inputs to the respiratory center. Initially, there is a very rapid increase that occurs too quickly to be accounted for by changes in metabolism. For example, movement of the limbs has a strong stimulatory influence on the respiratory center, which is what this illustration is showing. After the initial immediate increase in respiratory rate, there is a gradual increase that levels off within 4 to 6 minutes. The highest level of increase that can be performed without causing a significant change in blood pH, again, is called that anaerobic threshold. So that is what we are essentially looking at here. Okay, a few other modifications of ventilation include the activation of touch. Thermal and pain receptors can also affect the respiratory center. A sneeze reflex or a cough reflex, and an increase in body temperature yields an increase in ventilation. Same goes for a decrease in body temperature. And then last but not least, the effects of aging on the respiratory system. Your vital capacity and maximum minute ventilation will decrease, which is something we discussed earlier. Your residual volume and dead space will also increase. Your ability to remove mucus from respiratory passageways will also decrease with age, and gas exchange across respiratory membranes is also reduced. So that is the respiratory system. I hope you enjoyed it and have a lovely rest of your day. Make good choices and happy studying.